Good morning and good afternoon, everyone. This is Mahalea Marshall with the IQ Consortium Secretariat. On behalf of the IQ Consortium, I'd like to thank you for joining us this morning for the webinar entitled Challenges of Using Closed System Transfer Devices, CSTDs with Biological Products, presented by the IQ Biologics CMC Leadership Group's CSTD Working Group. Before I turn things over to Bharat Jagannathan, I would like to take care of a few housekeeping items. First, all audience members will be on mute for the duration of the webinar. You may submit questions at any time via the question module located within the control panel on the right-hand side of your screen. You may undock this module by clicking the small arrow in the upper right-hand corner for easier question entry. Please note that attendees may experience intermittent delays in slide transition or a disruption of audio if connected to voice over internet protocol due to local bandwidth demands. Once again, I'd like to thank you for joining us this morning, and I'll now turn things over to Bharat, who will introduce our speakers.
Hey, thank you, Mahalaya. So I am Bharat Jagannathan from Amgen, and on behalf of the Closed System Transfer Devices Working Group, I would like to welcome everyone to this webinar on the challenges of using closed system transfer devices, or CSTDs, with biological products. This will be a 30-minute presentation followed by a 30-minute Q&A session, so there should be enough time for a robust discussion. We have 3 speakers lined up for today. Twinkle Christian from Amgen will be kicking off the presentation with an overview of the technical challenges of using CSTDs with biologics. Twinkle is a process development scientist in the Drug Product Technologies group at Amgen. She has 15 years of industry experience in formulation development across diverse modalities and has led the advancement of multiple programs from early discovery to clinical trials and commercialization. Her expertise encompasses high concentration formulation and delivery of biologics, investigating protein-protein interactions and macromolecular crystallography. She's an adjunct faculty at California State University, Channel Islands, and she holds a master's in biochemistry and molecular biology. Prior to working at Amgen, she was employed at Alza and Baxgen. Twinkle will be followed by Isabella De Jong from Genentech Pharmaceutical Technical Development, who will be discussing both the technical and clinical challenges of using these devices. Isabella is a pharmaceutical development scientist at Roche Genentech with 20 years of experience in the pharmaceutical and biotechnology industries. She has worked on formulations and CMC development at all stages of product lifecycle for a variety of oncology, immunology, and ocular products. Prior to working at Roche Genentech, she worked at Shire Pharmaceuticals in formulation development. Isabella has a Bachelor of Science degree in Animal Science, Bioscience, and Technology from the University of New Hampshire. Finally, Alison Guy from Product Development Regulatory at Roche will wrap up the webinar with some regulatory considerations for CSTDs. Alison is a Director in Product Development Regulatory Affairs at Roche, where she works on shaping global regulatory strategies for early and late stage and marketed oncology products. Before joining Roche, she was a senior consultant at Paraxel Consulting, where she specialized in biosimilar development and manufacturing audit mitigation. Throughout her career, she has lectured on regulatory affairs at the Leslie Dane Faculty of Pharmacy at the University of Toronto and at Ryerson University. She has a Master's of Science degree in Neuroscience from the University of Toronto. With that, I will hand it off to Twinkle to start off the proceedings.
Thank you, Bharat. And thank you, IQ, for providing us the opportunity to present today. In the next 30 minutes, we will focus on the technical and clinical challenges of closed system transfer devices and briefly touch base upon the regulatory challenges. Next slide, please. So let's get introduced to the CSTD. So as per NIOSH definition, a closed system transfer device, or a CSTD, is a drug transfer device that mechanically prohibits the transfer of environmental contaminants into the system and the escape of hazardous drug or vapor concentrations outside the system. Shown below are some examples of CSTDs used in dose preparation and administration. Next slide, please. That should be the problem statement. Mahalia, can you go to the slide before this? Perfect, thank you. Okay, so the use of CSTDs is becoming more prevalent and in some cases mandatory for the preparation and administration of non-hazardous biologics in the US and globally. While the protection of the healthcare workers is paramount, understanding the potential issues and concerns to drug product quality and patient safety when CSTDs are used is of equal importance. Next slide, please. So why are CSTDs such a hot topic right now? So CSTDs are 510(k) cleared medical devices that have been on the market for more than 2 decades, aimed at minimizing exposure to hazardous drug vapors by creating a closed system for reconstitution or administration of hazardous drugs. NIOSH periodically releases a list of hazardous drugs and provides handling guidelines for preparation and administration of these drugs. In 2016 and 2018, biologics were included on the NIOSH hazardous list. The exposure risk of biologics is very different than that of small molecules, and this has been recognized by NIOSH in the latest revised draft of the hazardous drug list. In 2020, NIOSH excluded certain biologics based on their intrinsic molecular properties, and USP 800 guidance only apply to the drugs listed on the Table 1 of the NIOSH list. Even with these changes, however, we don't expect to see an immediate impact on the use of CSTDs with biologics, since CSTDs are already incorporated into the pharmacy practice and they are being used for non-hazardous biologics as well. Recently, there have also been several publications highlighting the potential issues and concerns to product quality and patient safety when CSTDs are used with biologics. Next slide, please. So CSTD is an umbrella term that is used for multiple components that can be used for compounding or preparation and dose administration. So as you can see on the slide here on the left, we are showing you devices that can be used for dose preparation. And these can be vial adapters, syringe adapters, or a syringe unit. For administration, you can have IV bags, spikes, and IV line connectors. If a clinic decides to use CSTDs only for dose preparation, they are more looking at the devices seen on your left. But if the clinic decides to use CSTDs for dose preparation and administration, you could be looking at about 5 components from a single brand being used before the dose reaches the patient. Next slide, please. So shown here on this slide is an example of how a CSTD can be used for dose preparation. As you can see in the picture, the vial is being cleaned and then a CSTD vial adapter, and in this case, the vial adapter has a skirt, is being attached to the vial. So the CSTD vial adapter skirt attaches to the neck of the vial, and at this point it is secure. It's almost impossible to take it off. Once it is secure, you can attach a disposable syringe, invert your vial, and draw out the contents of the vial. Next slide, please. Here is an example if a clinic would use CSTD for dose preparation and administration. So as you can see, you have a vial adapter as well that has been attached to a vial. Again, this one also has a skirt, so it's attaching it securely to the neck of the vial. In this case, we have a syringe unit. So the female adapter is glued on to this disposable syringe. The contents of the vial are drawn into the syringe, and then it is attached to an IV bag spike. Once the drug product is into the IV bag, the syringe unit is detached and you can connect the IV lines. At the end of the IV lines, we have 2 other IV connectors. You can see in the zoomed in picture, the female connector and the male lure. After this, the needle is attached and the dose can be administered to the patient. So at this point, we have used about 5 components from a single brand, and keep in mind that these CSTD components, either used for preparation or administration, are made up of different material types. So at any given point, your biologic could be in contact with different material types. Next slide, please. Okay, so let's take a look at the CSTD challenges with biologics. We have categorized them into 3 buckets. The first one is drug product compatibility. The second is clinical site preferences, and the third is device and regulatory guidelines. So first we will focus on the drug product compatibility challenges. Next slide, please. One more. Perfect, thank you. So CSTD technical challenges can be further divided into 2 categories. One is product agnostic and the other one is product specific. Product agnostic refers to the holdup volume and stopper coring, while the product specific relates more towards the drug product compatibility and the duration of product infusion. Holdup volume refers to the leftover drug product in a vial when a CSTD vial adapter is used. Stopper coring refers to the coring of the stopper material when a CSTD vial adapter is used. Depending on the technique, force used to insert the vial adapter, and the CSTD vial adapter spike, stopper coring can compromise drug product quality by generation of visible or subvisible particle formation. Product-specific challenges includes drug product compatibility and duration of drug product infusion. CSTD fluid path is composed of various material type, which may or may not be compatible with the biologic. Also, depending on your modality, the duration of infusion may vary, and it may lead to a longer contact time of the biologic with the CSTD material type of construction. This may cause incompatibility of the drug product, in turn compromising drug product quality. Next slide, please. So let's take a look into the holdup volume. So shown here are holdup volumes for various different CSTD brand, and as you can see, the holdup volume is variable. We have some CSTDs having a holdup volume around 100 microliter, which is similar to the needle and syringe control, and some CSTDs have a holdup volume in excess of 1 ml. So the green box is very similar to the holdup volume of a needle and a syringe, while the red box shown here has a holdup volume close to 1 ml, which is not acceptable, especially for oncology medications that may be in low volume SKUs to begin with. The blue box, or the gray box, as you can see on your screen, has acceptable holdup volume, though we had observed a really high subvisible particles, and on further analysis, we have figured out these to be silicon oil particles. So holdup volume can depend on several things: the CSTD geometry, the fluid path, and the operator technique. These may impact the ability to dose a patient from a single vial, especially in some biologics that have low fill volumes. This could also result in the need to use multiple vials to dose a single patient, which may lead to medication errors or an adverse impact on sterility. The holdup volumes are also issues that cannot be compensated by just overfill, as manufacturers are limited to a 0.2 ml overfill volumes by USP 1151. Next slide, please. Let's take a look at the next challenge, which is again product agnostic, extrinsic particles. So we see this challenge especially referring to the stopper coring. It's important to note that the CSTD vial adapter design differs by brand. Some vial adapters have plastic spikes, while some have metal spikes. Like I mentioned before, depending on the force and the technique used by the operator, it may cause stopper coring, and visible and subvisible particles may compromise drug product quality. Next slide, please. Here are some images that we have shown here of particles generated during stopper coring, and especially with a suboptimal stopper and a CSTD vial adapter combination. And at this point, I'm going to hand it over to Isabella for further technical and clinical challenges.
Thank you, Twinkle. Next slide, please. So what are the extrinsic particles that we've been observing? They generally fall into a few different categories. We've seen rubber stopper particles. The usage of some of the CSTDs resulted in rubber particles in solution, and there's a potential for the coring and fragmentation to end up in the dose solution due to the puncturing of the vial. We've seen CSTD-related material of construction, such as silicone polymetric plastics and polyethylene that can be shed into the dose solution during the extraction. We've also seen different lubricants that are used in the manufacturing in the CSTDs. For example, silicone oil-related lubricants. So in the figures below, for example, on the left-hand side, we did a study looking at the silicon concentration using ICP-OES. And in this study, 7 different CSTDs were evaluated and compared against preparations using a needle-free or a needle control. 2 independent preparations using separate drug product vials were used to prepare these solutions. And when we did the analysis, what we found is that in preparations using some of the CSTD brands, we saw detectable levels of silicon that was quantified. Additionally, we also saw variability between the 2 different preparations using the CSTDs. So for example, for CSTD number 3, we saw a lot of variability between the 2 independent preparations, indicating some variability between the CSTDs within a brand. Additionally, we did some microflow imaging of vials spiked with the CSTDs, and we see that the morphology of the particles and the droplets observed are consistent with what we would expect from a silicone oil. Next slide, please. So now I am going to talk about some of the product-specific challenges we've seen with using CSTDs. Next slide, please. So we've seen an impact on the subvisible particle formation with the preparation of dose solutions prepared using CSTDs. And again, in this study, we did preparations using 7 different CSTD brands compared against needle-free and needle controls. We did these preparations with both formulation buffer and 2 independent preparations of a MAB in 2 separate IV bags, M1 and M2. We did particle counts. 2 micron particles are listed on the y-axis, and the CSTD brands across the x-axis. In these preparations, the MAB and the formulation buffer were prepared and then injected into an IV bag, a 0.9% sodium chloride IV bag using the CSTD or the needle control, and incubated for 24 hours at 30 C, followed by simulated infusion using some of the CSTDs. And what we see once again is that there is an increase in some of there is a higher level of subvisible particles in some of the CSTD brands in comparison to the needle-free and needle control. And this is observed also in the formulation buffer preparations. But what we also see is a much higher level in the MAB preparations over the formulation buffer control. And another thing to point out is that even within one CSTD brand, we saw CSTD to CSTD variability in the subvisible particle counts if you look at the difference between the M1 and M2 preparation. Next slide, please. In addition to the increase in subvisible particle counts, we've previously observed higher amounts of soluble high molecular weight species, an increase in turbidity, and also the observance of proteinaceous visible particles in mAb drug preparations after dilution in IV bags. And here I have some information for one CSTD brand in particular. In the top left figure, we have the high molecular weight species count. We did a preparation in CSTDs in red versus a needle control, which is shown in blue. In these preparations, we again did an we assessed the high molecular weight species count at T0, after 24 hours at 30C in the IV bag, and then after post-simulated infusion. There are 10 independent preparations, and the data presented here is an average from those 10 preparations. And what we saw is that there's an increase in the high molecular weight species, particularly after 24 hours at 30C, when it's compared against a needle control preparation. And this was also observed after post-simulated infusion. Similarly, on the right-hand figure, we can see that we did a measurement of turbidity. Again, at T0, after 24 hours at 30C in the IV bag, and after post-simulated infusion. And what you can see is that there's an increase in turbidity in the preparations with CSTDs when compared against the needle control. And at the bottom, we have information about the visible proteinaceous particles observed. At T0, 9 out of 10 bag preparations demonstrated visible particles in the solution. After 24 hours, 8 out of 10 of the bags still demonstrated visible particles. And even after post-simulated infusion without an inline filter, 4 out of the 10 bags also demonstrated visible particles. And it should be noted that no visible particles were observed in control sample preparations with a syringe and needle. Next slide, please. Now I'm going to spend a little time talking about clinical site preferences and why CSTDs are selected at clinical sites. Next slide, please. So CSTD preferences depend on multiple factors: vendor contracts, generally pre-existing relationships with vendors, as well as the preparation of these contracts. Ease of use, there have been studies done and published where sites practice and use different CSTDs, and the preference of the hospital staff and the pharmacist can be a factor in the selection of the CSTD. And then there's region-specific preferences. Next slide, please. We did a market survey in 2019 looking at CSTDs used, and we did this in collaboration with Health Advances, and we looked at the CSTD survey demographics. This survey was a fair representation from hospital pharmacists and oncology nurses across a variety of settings. Most had significant experience with CSTDs. So you can see we looked at the different settings: academic, community, private, and then other types of institutions. We questioned 130 different sites. The majority of them were in the US, though we also surveyed countries in the EU and Australia. And what we found that most of the respondents had experience dealing with hazardous drugs. In fact, 75% of the respondents said that they handled hazardous drugs multiple times a day. And the people that were surveyed were also between 2 and 35 years of industry experience, demonstrating a broad range of experience in our survey. Next slide, please. So one of the questions we asked and we wanted to understand is how these sites define compatibility. Interviewees indicated that compatibility is defined in 2 different ways: the ability to mechanically attach the CSTD to a drug product file, and the potential changes in the drug quality due to the pressurization and potential aerosolization of the drug. Certain CSTDs might even retain a small amount of drug volume in the chamber, which can impact drug dose and potential efficacy if not accounted for. 50% of the respondents in our survey indicated that they rely on data from the CSTD device manufacturer to assess the CSTD compatibility. And we can see on the right-hand side that respondents from US, Australia, UK, France, Italy, Denmark, and Spain indicated that they check CST device manufacturing specs to determine if a CSTD is compatible. And other respondents indicated they asked the compatibility data from the CSTD device manufacturing, indicating that they're not necessarily coming to the drug manufacturer to determine compatibility when making selections on what CSTD to be used in their healthcare site. Next slide, please. And now I'm going to hand over to Allison to talk about device and regulatory guidelines.
Thank you, Isabella. So it's really interesting that the market survey revealed that healthcare providers are looking to device manufacturers to provide compatibility data in order to help them determine which device to use with which drug, because the device manufacturers are not obligated to produce this data as part of the approval process, especially not as compatibility was defined by the interviewees in the survey. So in the US, CSTDs are regulated by FDA as Class 2 devices. And typically, the device manufacturers use the 510K pathway for market approval. For the 510K pathway, if there's already a similar product on the market, the manufacturer really only needs to submit data to support substantial equivalence to the predicate device, so that they don't need to produce new data with their actual device. These devices also have a very broad indication statement, which usually just speaks to their ability to mechanically prohibit the transfer of environmental contaminants into the system and the escape of drug or vapor concentrations outside of the system, and how this limits individual and environmental exposure. So the labeling doesn't even get into compatibility with the products with which they're used. So what about the drug manufacturers? Of course, one could argue that they should be treated like combination products, but first of all, drug manufacturers, they're not the ones claiming that they should be used with their products. But even if they were, one of the gaps we're seeing is that the challenge is that there's no standard set of testing to be performed to support physicochemical compatibility claims between CSTDs and biologics. So not only is there a gap in terms of standards, but there's also a gap that where there's no definition about who should be doing the testing itself. And this is especially problematic when you consider that oversight bodies like NIOSH and USP have guidance documents recommending the use of these devices with hazardous drugs, some of which are biologics, and that most state pharmacy boards use USP as the benchmark for best practice, which leads to individual institutes broadly mandating the use of these devices for the preparation of biologics. So further collaboration is needed to bridge this regulatory gap. So if we go to the next slide, thank you. You can see that we really do need to have a more robust dialogue between drug and device manufacturers, but also with health authorities to ensure that we can clearly define who should be doing this testing and what the testing should be. So I will stop there. And before we go into opening the discussion, I'd like to acknowledge a few people who helped us with this presentation. So of course, the IQ Working Group and the IQ Secretariat for supporting the webinar, as well as the IQ leadership. So Bharat, I'll hand it over to you now to moderate our discussion.
Thank you, Twinkle, Isabella, and Allison, for the great presentation. We have quite a few questions, so maybe we can get right in. So the first question is, don't the devices need to conform to ISO standards? And that's probably a question for you, Allison.
Thanks, Bharat. Yeah, they do need to conform to ISO standards, and usually they need like an ISO 13485 certificate. But this standard only specifies requirements for a quality management system to be in place, and this stipulates that the organization needs to demonstrate its ability to provide medical devices and related services that consistently meet customer and applicable regulatory requirements. But as I went through a little bit earlier, there are really no regulatory requirements in terms of like compatibility testing for biologics.
Thank you. So the next question is probably for Twinkle or Isabella. Is there a concentration dilution impact when using CSTDs?
I can take that. So there are likely product-specific and formulation-specific differences that we recommend that should be assessed. But so far, we don't know if there are differences in terms of dilution. So it is recommended if a sponsor is testing CSTDs, it's recommended that they test the low and the high end of the dilution scheme or a worst case scenario. At this point, we don't have enough data to point to dilution dependent issues.
Okay, thank you. So Allison, question for you. What is the starting point to resolving the regulatory gap for CSTDs?
That's a great question. Okay, so I think we can all agree that we want to ensure that the safety of healthcare providers who are preparing our products are safe. Like, we want to ensure the safety of those people, but we also want to ensure that our patients are safe as well. And as you can see, that there are some technical considerations that Twinkle and Isabella went through at the beginning of the presentation that we need to take into consideration. So we do need to come together, I think, as drug manufacturers, device manufacturers, but also as health authorities to determine, how much regulation is needed, who needs to be regulated in this space, and what we need to do to ensure that we can protect the safety of healthcare providers and of patients.
Okay, thank you. So the next question is for Isabella. On the high molecular weight results you shared, was there any statistical significance between the use of CSTDs or not, red versus blue bars?
This was a real difference that we observed in those high molecular weight species. There was an average plus the SD based on the 10 independent IV bag preparations, and it was determined that this was a real difference between the syringe and the CSTD.
Okay, thank you, Isabella. The next question is also for you. Do we understand the scientific rationale for the high amount of high molecular weight species while using CSTDs? Is it more about material of construction incompatibility or any other physical issues like aerosolization?
I believe it could be both factors. I think there is product-specific regions, protein-specific regions. There could be, I think, mechanical stress that could be a factor, and there could also be material of construct. It's very difficult to determine given the variability that we see even between two independent preparations, and so that could potentially point to differences in the amount of lubricants that are released when using it, or variability in the manufacturing, but also could be very product specific. So I wouldn't say that we can say we exactly know why those high molecular weight species are formed, and generally would point to those factors: mechanical, material of construction, and then protein specific.
Okay, thank you, Isabella. So next up is a question on overfill volume. Since USP 800 recommends the use of CSTDs, is any work being done to advocate to USP for modifying USP 1151 to allow for greater excess volume / overfill for drugs that would require the use of a CSTD?
So I can start, and then I'll have other people jump in. So this is a question we've actually been talking about a lot at the IQ working group on CSTDs, and I think we might even have a webinar about it a little later on in the year. But it is kind of like a catch-22. It's like, if we want to accommodate the use of CSTDs, then there is a possibility we would need to overfill. But at the same time, there are guidelines about the limits on how much you can fill, and due to the variability in the holdup volumes across manufacturers, what is reasonable, and that's another area where we would appreciate some guidance, either from USP, from one of the health authorities.
Okay, thank you. All right, so next up is a $1 million question. Why are we using CSTDs for protein therapeutics at all? Twinkle, you want to take that?
Sure. I think, again, like Allison mentioned, that's another question we talk about a lot in our IQ working group. Biologics, by their properties, are not absorbed through the skin or through the nasal pathway. And most of them are definitely not hazardous. So again, that brings up the question as to why they're being used. It goes back to the original NIOSH, being all the drugs being anti-neoplastics, and then recently NIOSH did update that list in 2020. But that being said, CSTDs have been incorporated into the pharmacy practice, and at this point it's more of a blanket policy. So even with the NIOSH guidelines and the USP guidelines, it's almost a catch-22 where they are incorporated and clinics may be using it for all drugs coming their way, irrespective whether a molecule is hazardous or not.
Okay, thank you, Tomiko. The next question is for Isabella. Do inline filters mitigate the formation of subvisible particles?
Inline filters can mitigate, or they can clear the particle formation that is generated when using the CSTDs. So it is a good mitigation strategy, also given the variability that we see between the CSTDs, since you can't always necessarily predict these particle formations. So implementing the inline filters is a good mitigation, but as we're aware, perhaps inline filters can't always be used due to adsorption of the drug or the protein solution to inline filters. So care has to be taken when deciding to use inline filters to mitigate the CSTDs, and awareness of the dose solution and susceptibility to adsorption to the inline filters when deciding if an inline filter can be used.
All right, thank you. So the next question is probably for, well, I guess anyone can answer this. So many manufacturers make blanket statements for drugs in clinical trials that no CSTD may be used. We know that the fluid path of CSTDs vary and some may not present a risk to the biological because they use the same materials as a needle and syringe. Is there any recommendation how to avoid these blanket statements, or how to get the study sponsor to recognize not all CSTDs are the same?
Oh, go ahead.
Yeah, I can start maybe on this one. This is it's a great question. It's challenging. I mean, the sponsors recognize that CSTDs are desired to be used by clinical sites, and it's difficult sometimes given the different stages of clinical studies and how big the clinical study is to, one, be able to, we can make a blanket statement that it's not compatible, but it's difficult when working with the sites if they have a requirement to use a CSTD. It's also, without product-specific testing of the CSTD, it's challenging to agree to use of the CSTD and then keep track of which CSTDs are using at different clinical sites.
Yeah, and then if you wanna layer on top of that, you know, even if the materials of construction are the same to needle and syringes, there's still the issue of holdup volume, and especially in the clinic where you're trying to demonstrate efficacy, if you don't actually know the dose being delivered to the patients, it complicates whether or not you're not seeing the efficacy because the drug isn't effective or because patients weren't receiving the full dose.
One thing I would like to add also is that the material components for these CSTDs vary depending on the brand. And we're not just looking at one material type. There are multiple that are combined together to make a component. So it's also very critical to really know what the material type is of a certain CSTD. And then of course you have the product itself and the modality and product specific testing or information that you really need.
All right, thank you. So the next question is for Isabella. On the results of influence in subvisible particles, were other sizes analyzed instead of only 2 microns?
Yeah, typically we assess different subvisible particles, not just 2 microns in size, but for this, we presented the 2-micron particles. And that is typical part of standard assessment to look at 2, 5, 10, and 25 micron size.
Okay, thank you. So the next question is perhaps something that Twinkle can answer. Have CSTDs been used with synthetics, and do we see the same challenges?
I believe yes. They have been on the market for 2 decades and originally were meant to be used for synthetics. And the hold-up volume issue, I believe, remains irrespective of the modality. I think that relates to the amount of drug you have in a vial. If you have 10 mils and you're losing 0.2 mils, that's a different story. But if you have a low volume SKU to begin with, then you may have 1 mil or less than that, but you're losing 1 mil or more than half of your dose. I think that's where it creates problems in terms of not being able to deliver the correct amount of dose to the patient.
Okay, thank you, Twinkle. The next question is, again, it's kind of an open question. How can we expect the device manufacturers to perform compatibility testing? It makes sense for them to do studies like the coring studies or leaching of silicone oils, but the onus of compatibility testing should reside with the drug manufacturers. So I guess this was more of a comment than a question, but if Allison, Twinkle, or Isabella, do you want to comment?
Yeah, I mean, I certainly can appreciate that question, because you're right. How can device manufacturers know all the ins and outs of the IP of the drug product? But at the same time, this really just speaks to the regulatory gap, like who should be doing the testing, and if there are clear guidelines that, yeah, the device manufacturers will take this piece, the ones that were brought up, like stop recording, I think that's completely reasonable, or leachables and extractables, that's also reasonable. And then the drug manufacturer takes on another piece. I think that those are things that need to be discussed and decided as, you know, multiple stakeholders in this issue.
Okay, thank you. So the next question is somewhat related. Let's say you have data that shows your drug is not compatible with a specific CSTD brand for any number of reasons. What do you recommend the drug manufacturer do about that in terms of advice to clinical sites, especially after commercialization, where the drug manufacturer has little control over what clinicians use?
I think we've seen some precedents for if there's a true incompatibility with the material of construction, that's put exactly into the label of that drug product. So I think we have seen that, although I don't think there's any actual guidance around it yet. And I'm trying to remember the drug product off the top of my head. I think it's maybe Trianda, but it was incompatible with a very specific plastic. So in those cases, I think it's very important that that's communicated to customers and to prescribers at the end of the day.
Okay, thank you. So the next question, is, perhaps Isabella can answer this. If you have to perform a dilution of the product, would you still recommend a CSTD due to potential dose accuracy issues?
Sorry, and perhaps I'm not entirely understanding the question, but I'll try and answer. Dilution of the drug product prior to preparation of the final dose solution in IV bag. I mean, given the potential issue of being unable to accurately deliver the entire dose because of the extractable volume issue, you know, it causes potential concern. And then if there's additional, I guess, preparation prior to introduction into the IV bag, you could potentially compound that issue if you're doing a dilution steps prior to preparation of the IV. So you may compound the potential inaccuracies in the preparation of the dose solution.
Okay, thank you. So the next question is for Allison or Twinkle. Are there any efforts within this IQ group to interact with health authorities on this topic?
So I can start, and Allison chime in. We are looking into reaching out to health authorities. IQ has a specific group that can help with this to reach out to regulatory authorities. So the working group is planning to move towards this to get some guidance from the health authorities, especially for the CSTD, the testing, the compatibility, and maybe even standards for these devices.
Yeah, so I'm not aware of any ongoing efforts for industry to go together to health authorities, but I think it would be more powerful if we could get together to raise our concerns to see how we can go about solving or at least bridging these regulatory gaps.
Okay, thank you. So the next question is something that I can probably answer. Is the IQ working group planning on publishing any white paper based on the survey results and also the work done by the partner companies? The answer to that is yes. A white paper has just come out in proof of last week, and we can share that citation with this audience at some point. So the next question is for Allison. Is there a requirement to demonstrate the label volume can be extracted if the product is dosed based on the volume, so the dose is ensured?
Sorry, Brian, I'm not sure I quite understand the question, but I mean, currently all products are labeled for the extractable volume, and that's how much you need to be able to show was extracted, but those studies are done with needle and syringe. And so we get into this issue of the holdup volume that varies across. So even if you think you're extracting the whole content of the vial, when you go to administer into the patient with the holdup volume, you're not actually sure how much is the patient's actually receiving, which is the problem we're running into.
Okay, thank you. So the next couple of questions are for Isabella. Is it possible to share the actual survey results, not the summary?
No, unfortunately, I can't do that. But yeah, I can't share that right now. I know that there have been some other surveys that I believe have shared from Health Canada that are going to be published, but the particular one that I have, I can't share right now.
Okay, thank you. So the next question is for you, Isabella. Is in the survey, there were a great number of pharmacists and medical personnel who said they deal daily with hazardous drugs. Was the hazardous drug categorization according to the respondent opinion or a certain definition?
That's a great question. And I have to admit, I can't specifically remember what the definition of hazardous drug. It was probably, yeah, I'd have to go back and get back to that one. Sorry, I can't recall exactly where the definition of hazardous drug. It was probably due to the hospital pharmacist in oncology. We did not define hazardous drug for them.
Yeah, I think in that survey, the respondents defined it.
Okay, thank you.
Okay, so Allison, this is a follow-up of the previous overfill volume question. Who's responsible for the delivery losses when using non-CSTD devices for drug delivery?
Who's responsible for the losses for the non-CSTD products? Okay, so I mean, if because we're doing all the testing with needle and syringe, we have to be able to demonstrate that you can extract all of the labeled volume with needle and syringe, and we know generally how much what that would look like, and those losses are very small. So when you compare that with the range that we see of CSTDs, that range can go up to a full mil when a CSTD is used. So we're not seeing that with needle and syringe, so it's less of a concern. So although there is a very small volume loss, it's not the same as what can be seen when using these devices with the larger holdup volumes.
Okay, thank you. So Twinkle, perhaps you can take the next question. The holdup volume can be related to workflow techniques in addition to device characteristics. How would you propose considering these techniques that can be used for potentially mitigating holdup volume issues?
For most of the holdup volume comes from a specific CSTD brand. There's also geometry that is involved. At the 2019 workshop that we had conducted, there was some comments in terms of technique that is being used in the clinic as well. But at the end of the day, the holdup volume again is an issue, and that depends on what is in the vial. Basically, do you have a 1 ml fill? Do you have a 10 ml fill? Again, like I said, 10 ml fill, maybe not as much of an issue, but if you have a low volume SKU, it's definitely going to be an issue. And the way to get around it is maybe use an extra vial, but then you may run into medication errors. But if you're using a certain CSTD brand where you're losing a mil, the technique or the geometry, basically the geometry dictates the holder volume.
Okay, thank you, Twinkle. So the next question is probably for Allison. Would you recommend to register a specific CSTD with your product?
So we've thought about that, and through the survey, we actually learned that a lot of healthcare providers, even if a CSTD is co-packaged, have a preference for using the CSTDs that are stocked at their institute, mostly because they're trained on them. So it could be, even though that you go through the trouble of identifying an appropriate CSTD and then co-package it with your product, that at the end of the day, it's just being thrown in the trash and that someone's going to use whatever CSTD they're most comfortable with.
Okay, thank you, Allison. Isabella, this question is related to an earlier question. Can silicon oil particles introduced by CSTDs pass through the 0.2 micron inline filters? Is it a safe barrier for these liquid particles?
I think you'd need to do the evaluation. What we've seen is that when we use an inline filter, we can clear particles, and we haven't typically we haven't detected any issues when we do use an inline filter to clear those. So we do those assessments looking at the subvisible particle counts, and then inspection to make sure there's no residual silicon oil. I think that when doing these evaluations with CSTDs, it is a good idea to look at pre and post infusion with an inline filter to confirm for your product that you are clearing, you have acceptable particle counts.
Okay, thank you. So perhaps Twinkle can address this next question. In order to cover CSTDs, especially to add the information in the IFU, do you recommend any compatibility strategy that needs to be followed? For example, should we test out the most popular CSTD brands?
Well, right now you have 10 plus CSTDs on the market, and that number has only grown since the CSTDs were launched. So again, I think this is a really tricky question, and it goes back to what the individual company would like to approach this question. There are a couple brands that have a higher market share. That's one strategy. Another strategy could be, depending on your modality and you look at the material of construction for the CSTD, that was something that was suggested in the BPOC paper that came out last year. So, but at the end of the day, it still matters the compatibility of your specific product with the CSTD brand. I believe it will help the biological manufacturer if you know ahead of time what CSTDs are going to be used at a specific clinical site to make that decision.
Great, thank you. So we only have 5 minutes remaining, so I'll go through the next questions fairly quickly. So Allison, from a regulatory perspective, are companies including CSTD testing as part of in-use components testing for their IND filings? And is the data being included in INDs?
I don't have a line of sight to that, but it's not required. So this is where we're looking to get like more guidance from the different health authorities. But should we be doing this if we know they're being used in the clinic in order to make accurate recommendations about what we considered acceptable in the clinical trial?
Okay, thank you. So Twinkle, are there initiatives to interact with pharmacists and nurse associations via IQ?
We haven't done that yet, but that's something we have discussed as one of the goals for this year. So short answer, yes, we will be looking into that to make sure we involve the pharmacists and the nursing organizations.
Okay, thank you. Isabella, are particles from coring worse for CSTDs than with needle and syringe?
Yeah, we have seen, because of the geometry and the way that the spike for the CSTDs work, that we have seen that there's potentially more coring that comes with use of CSTDs. And in fact, getting alerts and questions from healthcare sites about the stopper and rubber particles when using CSTD is one of the first ways that we were alerted to the issue. It was actually the reporting of the particles in the vials and the rubber particle parts from the CSTD spiking that was really one of the first things that we observed or were notified of.
Okay, thank you. So, Twinkle, what would be your suggestions when addressing low dose accuracy situations while we have hold volume in the bag and line?
So I think that's something we account for when you do testing, especially with a needle and a syringe. That holds true even for any CSTD that is being used. But with the CSTD, you have the additional holdup volume that you lose when you use a vial adapter. So that's something that you need to account for. And like I mentioned before, clinics do have the option of an additional vial, but you are looking at medication errors depending on what that final dose is and how many vials you need to open up. So that's something that needs to be worked out ahead of time in collaboration with the clinic.
Is the question about hold up volume or is it about hold times? Is it about in-use stability?
I would imagine it's about hold up volume. They're asking about hold up volume in the bag and the line.
This hold time will refer to compatibility.
So that is unclear. So maybe we can move on to the next question.
Okay.
The issue for hold up volume particles, etc., should not be applied only to biologics. It's not only applicable to biologics. How has this been handled for others? So I'm guessing the question is, I'm kind of interpreting this here. How has the hold up volume issue been tackled for other modalities like synthetics?
Synthetics may have a higher fill volume, and I think the issue here, it's not necessarily about modality. I think it's more about the fill volume in a vial, and that happens to be with biologics and especially oncology products, which have a lower fill volume to begin with.
Okay, thank you. So I know we're almost out of time. The next question is, will the second presentation materials and Q&A be shared with the audience after the event? The answer to that is probably no. Just going through the list of questions here. So I have one final question on hold-up volume. For hold-up volume, there seems to be an assumption that the patient is ordered one vial versus a specific dose. The hold-up volume issues are negated when the dose is drawn up in a syringe. The only issue is if the dose volume in the vial is the exact ordered dose. So I'm not sure. I think that's just a comment. So Isabella, just one question for you. Do we understand the mechanism of visible particle formation? Are the high molecular weight species acting as a trigger or as a platform?
We think that it is related to the lubricants that come out, and so there could be potentially some coupling of the protein with the lubricant to generate these subvisible or these visible particles in the solution. So the lubricant is a factor, and the combination of the drug with the lubricant is a factor.
Okay, thank you. So maybe we can wrap things up with one final question for Isabella. How old are the silicon oil studies that were presented? Is that a legacy issue or has it been resolved?
Sorry, I guess to clarify, has it been resolved in terms of, yeah, I guess I'm just trying to understand the question. We're aware that it's an issue, and we have evaluated the silicon oil in the study, but all we can do is mitigate it. I mean, the variability in the silicon oil coming from the CSTD is something we need to have the CSTD manufacturers understand, and we can be aware and evaluate for the silicon oil or the lubricant in the vials and then mitigate it either by determining that our drug just isn't compatible with that CSTD. I mean, we talked about potentially we have talked about using inline filters as another mitigation strategy. I guess, yeah, hopefully that answered the question. It was a little bit unclear to me.
Okay, we are out of time. Thank you, everyone. This was a great Q&A session. There are still a few questions that remain to be addressed, but maybe we can figure out if there's a way to address these questions offline with Mahalaya. Thank you. I'll turn it over to you, Mahalaya.
We hope to see you at a next webinar. Have a great day.